So we've talked about how DNA is transcribed into messenger RNA during transcription, and that mRNA in eukaryotes is spliced up, and we get rid of introns and reattach exons in some order. And there is alternative splicing, which means we can get multiple final mRNA transcripts from a single gene, meaning make multiple proteins. And we've talked about translation, where we take that messenger RNA transcript and we translate it from the language of nucleotides to the language of amino acids using our bilingual molecule messenger RNA and a ribosome and the code that is on those codons, those 3-letter long codons on the mRNA. Now, one messenger RNA transcript can make lots of the same protein. In fact, very often it makes a lot of them at one time, right? Once the first part of a messenger RNA protein gets through a ribosome, and this ribosome is working on a later section of the mRNA, that first part of the mRNA can bind to a couple of subunits of a new ribosome, and you get what's called a polyribosome. And it looks like this, right? Here is our start codon, right? And our 2 subunits bind on, but you've still got lots of other ribosomes currently at different stages of protein production, and this one has just released because it's hit the stop codon. This is an actual electron micrograph of a polyribosome in action. So now we have a polypeptide with a primary structure within an amino acid sequence. But that's not the end of protein production. That primary sequence, that primary structure, has to then allow for a secondary structure, right? Remember the helix or the pleated sheet, and a tertiary structure where it folds in those crazy 3-dimensional ways based on hydrogen bonding and hydrophilic and hydrophobic regions. And sometimes you will actually put different polypeptides together, right, to make these giant proteins. A lot of this modification might happen in the Golgi apparatus, as we've talked about. And we also know that some proteins will wind up just in the cytoplasm, and some proteins will wind up either in the membrane or leaving the cell, being exported. Those membranes bound or sort of exit bound proteins need to begin their lives in the rough endoplasmic reticulum, which means that's where you'd end up on a ribosome that's dotted in the rough ER. Well, there's nothing different about the ribosomes between free-floating ribosomes and rough ER ribosomes, right? What dictates where they go is actually part of the mRNA. Right? So there are, if you take a look, something called a signal peptide on the mRNA that will mark it for rough endoplasmic reticulum destination rather than being free-floating. That signal peptide binds to a signal recognition particle, which brings it over to the ER, probably using microtubules. So this is what that looks like, right? We have a ribosome, and our mRNA codes for one of the first, sets of amino acids is a signal peptide, which will bind to a signal recognition particle, which will truck this whole thing over, boop, as it's being made into a rough ER, right? So actually, the first part of the polypeptide has been made before it even gets to the rough ER, but the rest of it will be made into the rough ER, and bam, we have a protein that's now in the rough ER, and it may go on to be cleaved or bound with another polypeptide and wrapped up, or it might be, might have a sugar added or a fat or something like that. All of these things are possibilities. Well, what happens when things go wrong? When the DNA has had a mutation, right? Well, mutations can come in a number of different forms. Point mutations are mutations of a single letter, right? A single base pair, right? An A becomes a T or becomes a G or something like that. And sometimes that may not matter much at all, right? If because there's some redundancy and some of the same RNA codons, or some different mRNA codons will code for the same amino acid, right? If your mutation makes a change but still codes for the same amino acid, right, you'll never even know there was a mutation. But sometimes even just a single point mutation can cause a big difference. So this is the template strand and the mRNA for wild type hemoglobin, right? And this GAA, this is just a piece of it, right? This GAA would code for glutamine. This CTT changes to a CAT in some people, and this single point mutation changes our GAA to a GUA and changes that glutamine to a valine, and all of a sudden the protein structure of hemoglobin is different, and you end up with the symptoms of sickle cell anemia, which we've talked about before. Now, these small point mutations can either be a substitution, where again, just like in the last example, an A becomes a T or a G, or a C becomes a T or a G or something, or they can be a deletion, or you don't substitute anything for that A, you just take it out, right? Or you add an extra G in there, right? An insertion. Those substitutions can be a big deal, right? But not always. Again, there are silent mutations because of the redundancy of that mRNA code. Missense mutations code for a different amino acid, not the correct amino acid. So in the last example we talked about, this is a missense mutation, right? It's a point mutation, it's a substitution, T becomes an A, and this A now means we code for a different amino acid. That is a missense mutation. But it can be worse. It can be a nonsense mutation if the new amino acid you code for, right, isn't an amino acid at all, but a stop codon. So here, right, we've started with the same wild type. Here, we have changed, instead of coding for a lysine here, right, a single point substitution, point mutation, gives us a stop codon. Now, these missense mutations can be terribly destructive because then you don't make a huge chunk of the polypeptide, and clearly you're going to lose function there. Now, this is more likely to happen when you get an insertion or a deletion in what's called a frame shift mutation. So if you just swap a single nucleotide, you're more likely to have either a nonsense mutation or a missense mutation where you just get a single different amino acid that you code for. If you add or subtract or delete a nucleotide, then you shift the whole frame, because we're reading this three at a time. So imagine we have, for instance, a sentence, and it says the cat ate the rat, right? That's pretty easy to understand, and we have all three-letter words, just like we do in DNA, right? A triplet code of nucleotides. Well, imagine I deleted the E, right? But we still read it 3 letters at a time. So it doesn't just say, cat ate the rat, which, you could still understand what that meant, right? It hasn't changed the meaning much. But it would be at tet her at, which is clearly nonsense, right? And so because this deletion shifted all of these over in the way that we read them, right, it's called a frame shift mutation. And frame shift mutations often have much bigger impacts than a simple substitution, right? Whereas if I had said, their cat ate the rat, right? This would be a substitution. Again, the sentence is substantially more similar. If this were DNA, the protein would be substantially more similar, right? The only amino acid that may have changed is this one. May not even have if it was a silent mutation. Here, every amino acid after our insertion or deletion is changed. Now, this can also happen if we add one in. So maybe I add in a G. Now I say ka, tat, et, erat, right? Again, an addition also changes everything after it. We've shifted the frame in which we're reading this, and so it's called a frame shift mutation. And not only might these frame shift mutations cause different amino acids later, which mean they can entirely change what the protein looks like, but they often lead to things like stop codons, right? Shortly after where your insertion or deletion was, which will make this a nonsense mutation, right? Meaning you lose all of the rest of those amino acids. They're not even just different, they are gone. Anything that can cause these errors during replication or repair or recombination, if we're talking about germ cells or sex cells, we call these mutagens. They cause mutations, and these could be chemical agents or physical agents, things like UV light. And if these cause mutations often in the genes that govern the cell cycle, we call them carcinogens, right? Carcinogens are just mutagens that are more likely to cause mutations that result in cancer. Okay, let's do a quick review of gene expression. So we start with the information, which is locked in the DNA inside the nucleus, at least if you are a eukaryote. Transcription is where we make not a copy of the DNA, right, a complement of the DNA, but containing the same information in a messenger RNA transcript. If you are in a eukaryote, that messenger RNA transcript will be processed. You will splice out what we call introns. You will add a poly A tail and a cap made of guanine on the other end. And then that will leave the nucleus and find a ribosome where we can go through the next part after transcription, which is called translation. Translation, translating from nucleic acids to amino acids, is going to involve tRNA, which is our bilingual molecule. Right? It has an RNA anticodon, which will match up with the codons on the mRNA. Right? And it has an amino acid on the other side, thanks to this enzyme here. They will come together at the ribosome. The tRNA will enter. It will arrive at the A site. At the P site, it will form a peptide bond between its amino acid and the chain that you've got going. And then it will exit to the E site, and another one will follow behind it. And you end up getting a chain of amino acids. Which amino acids in which order? Well, that's determined by the codons here, because they dictate which tRNA molecules come in. And those codons are originally dictated by your DNA. And that is how DNA codes for proteins, and that is how DNA dictates the traits that you have in your cells and in your body. Now, in the next chapter, we will discuss how we regulate that DNA, because clearly you have the same genes in every single cell throughout your body, but all of the cells throughout your body are not exactly the same. Well, how do we make that happen? That'll be for chapter 18.